Now we're going to get into some basics regarding hereditary genetics. We're going to talk about different types of inheritances, and then we're going to relate this to chi-square, and then eventually you're going to be set up so that you can do the fruit fly virtual lab. Now, since we just finished talking about meiosis, we have recently covered some of the basic ideas that you have to understand before we start doing Punnett squares and talking about the types of inheritances. We've mentioned that if we have diploid organisms that reproduce sexually, then these organisms are going to have two alleles for most traits, and they get one from each parent, one from mom and one from dad. When that organism goes to reproduce, it's only going to pass on one of its alleles for the trait. Now I want to mention a term that I'm going to use often, the allele. Here I have a pair of homologous chromosomes. So again, one is from one parent, so let's say this one's from dad, this one's from mom. So again, since these chromosomes are homologous, then that means they're going to have matching genes. So in this location, there's a gene for flower color, and in this location, there's a gene for flower color as well. But you can see that there's different codes or different variances at these genes. This one has a code or an allele for purple flowers, and this one has a code or an allele for white flowers. So when I say the alleles, what I'm talking about is the different variances of the same gene. So for example, for a gene for height, there could be an allele for tall and an allele for short. So again, there's two different alleles for the same gene. To represent the different alleles for the same gene, we use letters of the alphabet. So for the purple allele, I would give it a capital P, and I do that because it is dominant to white. For the white allele, I give a lowercase p. Lots of times I put a line over that lowercase p so you can tell the difference between a capital and a lowercase p. And so white is recessive in this case. So the genotype for this organism is going to be capital lowercase. So when we're writing a genotype, we're writing the symbols for the genes or the alleles that this organism possesses. And then its phenotype is what it looks like. So when we look at this plant, if it has this genotype, then we're going to see that it has purple flowers in this case. So phenotype refers to the physical characteristic. Genotype refers to the genes, the letters that represent the alleles that this organism possesses on its chromosomes. So these ideas represent just some basic ideas regarding genetics. And these ideas were established by the father of genetics, Gregor Mendel. So these ideas that I just went through were actually his ideas. What Gregor did is he crossed pea plants and he looked at the offspring and the phenotypes of the offspring and recorded this data, and he was able to figure out how certain traits were passed down from generation to generation. So he gets the credit for these ideas. He determined that each person, actually each plant or each organism, has two factors for every trait. He called them factors. We call them genes or alleles for every trait. He also figured out that these two factors will segregate. That means that only one factor is passed on to the offspring from the parent, and then the other parent will contribute another allele or another factor for the trait. So he determined and came up with the law of segregation, and we've already talked about that. He also determined that the passing on of a trait happens independently of other traits. So he saw that flower color and the passing on of those different traits really had nothing to do with the passing on of how tall a pea plant was. So this is the law of independent assortment that we've already talked about. Now, he was working with very simple traits in pea plants, and these traits were showing complete dominance. That is, there was one allele that was dominant to another allele. And so he thought that for every trait, there was always going to be one factor or one allele that was dominant to the other. So he came up with this law of dominance. So since he was working with pea plants, and a lot of those traits are very simple and just determined by alleles that show complete dominance, he didn't see other types of inheritances in the traits. He didn't see things like incomplete dominance or codominance. And if he had seen those, then it probably would have really thrown him off. So it was kind of luck, I guess, that he picked pea plants to do these genetic experiments with, because they show very simple inheritance patterns, like inheritance patterns that are associated with complete dominance. Now, Mendel had no idea about meiosis and the fact that we are packaging up these chromosomes in gametes and passing one of them on to the next generation in eggs and sperm cells. So he figured this out. He figured these ideas out with no knowledge of this diagram. But it's easy to see his law of segregation and a law of independent assortment in these diagrams, because we know this is what happens during gamete formation during meiosis. I think students have a hard time trying to explain the law of segregation and the law of independent assortment because they sound very similar to one another, but it is something that you need to be able to do. So again, the law of segregation means that these two alleles that you possess for a certain trait, they're going to be segregated into different gametes. So what that means is that this big R and the little r are going to end up in different gametes. So if we look down here, then we're going to see that there's only going to be either a big R or little r in each of these different gametes. So that is what we mean by the law of segregation. You will only pass on one of your two alleles to the next generation. The law of independent assortment can be demonstrated in this cell in metaphase 1 here. Whenever these chromosomes line up, they line up independently of one another. So the next time these chromosomes line up during meiosis, we can see a different lineup. We can see maybe the one with the capital R on this side and the lowercase on this side. And these are lining up just independently. They're not lining up with a certain R chromosome. So that's what we mean by the law of independent assortment. Before I move on, make sure you know these different ideas of Mendelian genetics, and also be able to talk about Mendel's 3 laws of inheritance: the law of independent assortment, law of segregation, and the law of dominance. So let's take a look at some crosses of traits that follow Mendelian genetics. So basically, we're just going to look at some traits that show complete dominance. If you have a trait that shows complete dominance, then that means that you have one allele that can dominate, mask, or cover up the other allele. The allele that is always expressed is called the dominant trait. The trait that's being masked is called the recessive trait. And again, we're going to use the first letter of the dominant trait, and we're going to make it capitalized to represent the trait. So if purple is dominant, then we're going to use that capital P for purple. And since white's recessive, we're going to use the same letter of the alphabet, and we're going to make it lowercase. Again, the line just indicates that that's a lowercase p, because sometimes some letters of the alphabet look the same whether you write them capitalized or lowercase. So if an organism has 2 chromosomes, and both chromosomes contain that allele for purple, then they're going to have a genotype that's big P, big P. Now, another word for this genotype is homozygous dominant. If I say an organism is homozygous dominant, you have to know to give them a capital letter as their genotype. I can also say that they're purebred for purple. That means that of their 2 codes, they're pure for purple. That's all they have. They have 2 purple alleles. Now again, phenotype is what they look like. So if you have a pea plant that is purebred for purple, or big P, then obviously they're going to be purple. Next we have an organism, and they have a code for purple and they have a code for white. So we say that they're big P, little p, and the fancier way to say that is they're heterozygous. Homo means the same, so their 2 letters are going to be the same. Hetero means different. So if you hear that an organism is heterozygous, you have to write 2 different letters for their genotype. I can also call them a hybrid. That means they're a combo of 2 alleles. They have the dominant one and they have the recessive one. Since purple is dominant to white, then the phenotype of the heterozygous individual is also going to be purple. If you have 2 codes for white, then we say that you are homozygous for white. In a genetics problem, if they say an organism is homozygous white, and you know white's recessive, then you have to know to write little letter for their genotype. We also can say they are homozygous. Homo means the same. So you have to give them two of the same letters. But we have to say recessive so that you know they have the recessive alleles. They're little letter, little letter. This is the only situation where you're going to end up with offspring that show that white phenotype. I know that you've seen Punnett squares before, and Punnett squares are just used to predict the phenotypes and the genotypes of the offspring if we know the genotypes and phenotypes of the parents. And when we see a Punnett square that looks like this, then we're doing a monohybrid cross. So basically, we're just analyzing one trait. Like for this example, we're going to use height. Height shows complete dominance in pea plants. Tall is dominant to short. Since tall is dominant to short, I'm going to use a capital T to represent the allele for tall, lowercase t to represent the allele for short. So let's go to these problems so that you can practice reading through them and making sure that you can figure out the genotypes of the parents before you start working through a Punnett square. It says cross a homozygous tall plant with a purebred short plant. So that means that you have homozygous tall, purebred short would be little t, little t. Now I filled in these Punnett squares to save time. But what this Punnett square is showing you is that this parent sometimes passes on a big T. Remember, each organism is only going to pass on one of each allele to the next generation. Sometimes it can put a big T, or the allele for tall, in a gamete. Sometimes it puts this one into a gamete. So it can pass on either a big T or a big T. And then this one is showing you that this parent is either going to pass on a little t or it's going to pass on a little t. And then you bring the letter down, take the letter across. So in this box we have big T, little t. I like to write the capital ones first. Now sometimes you're asked for genotypic frequencies or ratios. Sometimes you're asked for phenotypic frequencies, phenotypic ratios. You need to pay attention to what they're asking for. So if they're asking for a genotypic frequency, then they want the letters, they want the genotypes. So in this situation, you'd write 4 out of 4, big T, little t. Now I'm going to skip doing the ratio for now because I'm going to come back to that after I do the ratios for these other ones. For the phenotypic frequencies, we are going to have tall, 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 tall. And not everyone will write in the phenotypes there, and you don't have to, but for some of you, it might be handy for you to write in the phenotypes, and then you're not going to make simple mistakes. So the phenotypic frequencies would be 4 out of 4, and you write tall. Moving on to the next example, cross two plants, both heterozygous for height. So you would know that one parent is big T, little t, the other parent is big T, little t. Both are heterozygous. And I have it set up for you already, so this parent can pass on the big T or a little t, and this parent can pass on the big T or a little t. Bring them down, take them across, so we know that this box would end up being little t, little t. Genotypic frequencies. We have 1, we have these two are the same, and this one's different as well. So we have 1/4, big T, big T. 2 out of 4, big T, little t, which we can condense this down to 1/2. I like to leave it as a 4. You're going to see why here in a second when we figure out ratios. And then I have 1/4, little t, little t. Now let's go ahead and do ratios. When you do a ratio, you have to make sure that the denominator, the bottom number, is the same in all these fractions. And then you just take the top number. So we would write, we have 1 of this genotype to 2 of this genotype to 1 of this genotype. So that's how we do our ratios. If we go back over here, think about it. We have 0 out of 4, big T, big T. We have 0 out of 4, little t, little t. So this genotype can be shown as a ratio like this. We could write 4, big T, little t, to 0, to 0. And again, the denominators are the same. You just take the top numbers, 4, 0, and 0. Now let's go down here and look at the phenotypic frequencies. So in this case, we're going to have tall, 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 and this one's short. So this is to show you that just because we write 1/4, 2/4, 1/4 for the genotypes, that doesn't mean that's the same fractions that you're going to get down here for the phenotypes, especially if a trait shows complete dominance. We would have 3/4 tall, 1/4 short. And to change that into a ratio, we just check that the denominators are the same, which they are. We take the top number, so we have 3 tall to 1 short. Now going to this last problem, it says cross a hybrid. You have to know hybrid means hetero with a short plant. I have that set up here. I'm just going to jump down here to my phenotypic ratio. We can see that we get 2 tall to 2 short. This can be condensed down to a 1. So you can either report the frequencies as 2/4 tall, 2/4 short. So that gives you the 2 to 2 ratio. Or you can condense this to 1/2 and 1/2. So then we take the top number. And again, it would be 1 tall to 1 short. Now, the thing with doing these problems is that you need to be so good with them that you pretty much have expected ratios and frequencies memorized. What they're going to do in AP problems for this unit is they're going to show you what we got in the offspring, and you have to work backwards. You have to figure out what were the 2 parents that we crossed. And you have to do this very quickly because you're working on a timed test. Whenever I see this and I see that I'm getting all tall to 0 short, then I have different possibilities for the parents. I may be crossed a big T, and a little t, little t. That would actually give me all tall offspring. Or I could have crossed a big T, and a big T, little t. Because that would actually give me all tall offspring as well. When I look at this generation, and I see that we got that many tall and that many short, your mind has to do the math pretty quickly to figure out this is actually a 3 to 1 ratio. In this situation, we crossed a parent that was heterozygous with another parent that was heterozygous. And then whenever I look at this, I can tell, oh, that's a 1 to 1 ratio. Those numbers are pretty similar to one another. One way to get that is if you cross a heterozygous individual with one that's homozygous recessive. And that would give you half tall, half short, or 1-1 ratio. So again, do plenty of these problems until you basically have these ideas memorized. Cross 2 heteros, it's a 3 to 1 generation. You cross a hetero and a homozygous recessive, it's going to give you a 1-to-1 ratio. Again, if you have these ratios memorized, then problems like this are going to go very quickly. So go ahead and hit pause and try to answer it, and then come back and see if you got the correct answer. When a tall and a short plant were crossed, approximately 151 of the offspring were tall and 142 were short. So that's telling me right there, there's a 1-to-1 ratio. That's telling me that this individual was heterozygous, big T, little t. And of course, the short one could only be little t, little t. That would give me a 1-to-1 ratio, about half tall and half short. So 2 of the tall F1 offspring were crossed, and it resulted in F2 offspring. And we got about 156 tall, 53 were short. Determine the genotype of the tall plant in the P1 generation. Well, I already figured that out, but I'm just going to show you the second cross to give us this F2 offspring. It says 2 of the tall F1 offspring were crossed. Now, one thing to point out is the original generation is called the P1 generation. And then their children in the box, they're going to be the F1 generation. So we ended up with, for this generation, there was the tall ones, and here are the short ones. And the problem says we crossed 2 of these tall F1s. Well, here they are, right here. So that is why we got a 3-to-1 ratio. Because we've got these 3 were all tall, and this one was short. Before we go on to the other types of inheritances that don't fit the laws of Mendelian genetics, let's take a look at a test cross. If we're doing a test cross, then we have an organism that has an unknown genotype. And we're trying to figure out what that genotype is. When we do a test cross, it is always going to be an unknown. So basically, we don't know if it's big T, or big T, little t. We can tell it's tall, but we can't tell its genotype by looking at it. We take these unknowns and we always cross them with a homozygous recessive individual. That is what a test cross is. So if we have a pea plant that's tall, we know that its possible genotypes are this and this. And we're trying to figure out if this pea plant is purebred for tall or if it's heterozygous or hybrid. So a scientist is going to choose a plant to cross with this tall plant, and they're going to choose one that we can definitely determine its genotype. We definitely know a little t plant is a short plant. So if we cross an unknown, I'm going to put unknown up here. You know it has one capital T, so I'm going to put a capital T there, with this one that we do know, a little t. So let's say we end up and our offspring are tall, and about half of them are short, then we know exactly what the unknown was. The unknown was heterozygous, because we got half tall and half short. If all the offspring ended up tall, then this unknown tall plant was probably homozygous recessive. But since we got half tall, half short, then that unknown was heterozygous for the trait. So if you see a problem like we have here in problem 2, and it says that we have an unknown, discuss a way we could determine the genotype of a tall F2 plant without sequencing the plant's DNA, then you have to know we're going to do a test cross. But just saying that we're going to do a test cross is not going to be sufficient to get you enough points on the short answer. You need to explain that we're going to take this unknown and we're going to cross it with a homozygous recessive individual. We're going to take a look at the offspring, look at the phenotypes that arise in that generation, and help us determine whether the parent was homozygous or if they're heterozygous for tall. Now, these next types of inheritances that we're going to get to are known as exceptions to Mendelian genetics. They do not follow those rules or those ideas that Mendel developed. So these other ones that I know I've taught you before, like incomplete dominance, codominance, multiple alleles, like with blood types, sex-linked traits, these are types of inheritances that do not fit Mendelian genetics. In addition, we talk about there's some traits that will develop, and it's due to the genes found in the DNA on the mitochondria or the chloroplast. So that is what we mean by non-nuclear traits. They're not going to follow those ideas or those laws regarding Mendelian genetics. So if a short answer asks you to discuss a trait that does not follow Mendelian genetics, then pick one from this list above and talk about it. And again, pick one that you know a lot about. In your review book, they might talk about epistasis and pleiotropy, and they're not going to show the ideas of Mendelian genetics either, but I'm crossing through them because it's stated in the AP curriculum guide that those concepts are beyond the scope of the AP exam, so they're not going to be on the AP exam. You can read about them if you want. They're pretty simple to understand if you ask me, but you're not going to have questions about epistasis and pleiotropy. On the rest of these pages, we're going to talk about these exceptions to Mendelian genetics. So the first one is when traits show incomplete dominance. So this is an inheritance pattern where that heterozygous individual produces an intermediate or a middle of the two alleles for a certain trait. Sometimes this is called a blended phenotype. Now, when I symbolize the alleles for a trait that shows incomplete dominance, I like to use capital letters and primes. And the reason why I do this is because if I write big R, little r, you might think that whatever this one codes for will cover up this one. But it doesn't. They're incomplete to one another. So that's why I like to write them like this. And then as I'm working a problem, when I see my letters written like this, I don't make that mistake in thinking one is going to cover up the other one, especially when I'm working with a trait that I'm unfamiliar with. And you're going to be unfamiliar with some of these traits you're going to be working with in AP problems. An example of a trait that shows incomplete dominance is snapdragon flower color, because we have individuals that will end up red, individuals pink, and individuals that are white. So if we see a heterozygous individual or one of these phenotypes is an intermediate or is a middle of the other, then this is screaming incomplete dominance to you. Another example is with cholesterol levels. So people can have alleles for low cholesterol. I'm going to use a capital H to represent that. High cholesterol is H prime. If they're homozygous for low cholesterol, then they have two codes for low cholesterol, and obviously low cholesterol will be their phenotype. If they're heterozygous, then they have a moderate amount of cholesterol. And if they're homozygous for this condition called hypercholesterolemia, then they're going to have high cholesterol. Again, these traits that we just went through, they're exceptions to Mendelian genetics. They do not fit the law of dominance. So that's why they're an exception. Let's take a look at some crosses and some of the different ratios that you need to memorize. So it says cross a red flowering plant with a white flowering plant. So I'm going to use R to represent red. So this is a red plant. This would be my white plant. So there's just two alleles for this trait, an allele for red, allele for white. Notice there's no allele for pink. There's just two alleles, one for red, one for white. So when we do this Punnett square, we can see that all are going to end up pink. If I cross two pink flowering plants, then I would have to know that pink is the intermediate. So the pink ones are going to be heterozygous. They're going to have an allele for red and allele for white. Both of them are pink, and both of them are heterozygous. When we do that cross, then this is our phenotypic frequencies that we're going to see in the next generation. Cross a pink flowering plant. Again, pink is the intermediate, so you have to know that pink is going to be R prime with the white one, and this is what we're going to get. Again, to practice ratios, as long as the denominators are the same, you take the top number. So this would be 1 red to 2 pink to 1 white. But again, this is more like the problem that you're going to see on an AP exam. They're going to give you what the F1 generation looked like. So they're going to show you the phenotypes of the offspring, and you're going to have to figure out what were the genotypes of the parents. And they're not going to give you results like 1 was red, 2 was pink, and 1 was white. Because whenever we do crosses between individuals, we just don't get 4 offspring. From plants, we can get hundreds, if not thousands of seeds that we can plant, grow, and then have offspring there to analyze. So here we have a bunch of offspring, but you should be able to look at these two numbers right here and note that it's a 1-to-1 ratio. And if you've done enough of these problems, then you're going to be able to quickly decipher that we ended up with a pink parent. They were hetero. And when we crossed them with a white parent, then that gives us what we see up here, a 1-to-1 ratio between pink and white. If we end up with offspring and they're all red, this is an easy one. We cross 2 reds, and they were both homozygous for red. And then if we see these results, we have all 3. And if you notice, this is a 2-to-1-to-1 ratio, which is what we got here. I just have it written in a different order. It's still a 1-to-2-to-1 ratio, no matter what order I write it in. So we would have to note that we crossed 2 heterozygous individuals. We crossed 2 pinks. They were RR prime. Okay, so another type of inheritance that does not follow the rules of Mendelian genetics is codominance. In codominance, we have both alleles being expressed. One cannot mask or cover up the other one. In this situation, we're going to use 2 different capital letters to represent these 2 different alleles. Now with blood types, there's actually a whole bunch of different proteins that we find on the surface of red blood cells, not just the A, B, O blood types that you're talking about. There's, like I said, there's lots of other groups of proteins. Like there's the RH factor proteins we find on the surface of red blood cells. There's the MN blood group. And we actually have to type a lot of these and consider them whenever we're figuring out what blood can be donated to you if you go into surgery. It's not just typing that A, B, O blood type. There's other things we have to worry about too. So the MN blood group shows codominance. What we mean by that is someone that is homozygous for M. When we take a look at their red blood cells, then they have a marker on their surface, and I'm just going to put an M there. That's all they possess, is they have this M marker. Organisms that are heterozygous, on their red blood cells, we see the M marker, and it looks just like this one over here. But we also see what this one codes for, because both are expressed. So we see M markers and we see N markers. If someone is homozygous for those N markers, then we just see N markers on the surface of the red blood cells. So again, this shows codominance because when you have a heterozygous individual, you see what both alleles code for. They're codominant. They both show up. Another example is with coat color in some organisms. If they're homozygous for red, they're going to have red hairs. If they're homozygous for white, they're going to have white hairs. If they're heterozygous and these 2 alleles are codominant to one another, then you see both red and white hairs, and that condition is called roan, or you say they have a roan coat. Now, I want to go through the sickle cell anemia example, because sickle cell anemia, it does show codominance, but maybe not in the way that we're used to or we've been taught before. And I want to point out the way that they are symbolizing these alleles that code for normal hemoglobin or the sickle cell abnormal hemoglobin. See how they're writing their alleles here? It doesn't matter necessarily how you write alleles. You just have to remember what these different symbols mean and whether they're codom to one another, incomplete dominant, or complete dominant to one another. So if an individual is homozygous for normal hemoglobin, what that means is inside of their red blood cells, their hemoglobin all looks like this. It's normal. And that's all they have is normal hemoglobin. And their red blood cells can maintain this normal shape. If an individual is heterozygous, they have a code for normal hemoglobin and this abnormal mutant hemoglobin. And this is what it looks like inside their red blood cells. They have the normal, and it looks just as normal as this hemoglobin, but then they have hemoglobin that does this. It becomes clumped whenever there's low oxygen. And that is going to cause this red blood cell to turn into a sickle shape. And it only forms a sickle shape whenever it's in tissue that has low oxygen. If an individual is homozygous for sickle cell anemia, then all we see inside of the red blood cells is hemoglobin that becomes clumped. Whether there's plenty of oxygen or low oxygen, it's all like this. And this red blood cell is always going to look like that. So again, this is a little bit different than I know how you were probably taught in your previous class. But this trait does show codominance, because if you're homozygous for normal, all you have is normal. If you're heterozygous, you see both. You see both types of protein. You see the normal, and you see the hemoglobin that leads to the sickle shape. And again, these traits that we just went through showing codominance, they're exceptions to Mendelian genetics because, again, they don't follow the law of dominance. We don't have one allele that is dominant and the other one that is recessive all the time. Another exception is whenever we have a trait that has multiple alleles. Now, so far, everything that I've gone through for a certain gene, for a certain trait, there's only been two alleles. So there was normal hemoglobin, abnormal hemoglobin. There was tall, there was short for plant height. There was purple, and there was white. Here's a situation where in our population, there's 3 different alleles. Now, out of these 3, you still only possess 2, but there's 3 different alleles found in our population. So blood type has 3 different alleles. There's an allele for A blood. This is A blood. This is how I'm going to write the allele for B blood. And this is the allele for O blood. And again, it doesn't necessarily matter how you write these alleles for these different traits, but if they give you a symbol, I would go with exactly what they give you so you don't get confused when you're trying to solve this problem. Whenever you have multiple alleles, then these alleles might be complete dominant to one another. 2 of them might be incomplete to each other. 2 of them might be codominant to one another. So they're going to have to give you this information. In this situation with the blood type, both A and B alleles are dominant to O. So if you have a copy of A, which you have here, and you have a copy of O, you're going to have A blood. That A allele is dominant to that O allele. Now, if you have A and B together, then they are codominant to one another. So here's a situation where you have an A allele and you have a B allele, and your blood is AB blood. So basically what's going to happen in this situation is you're going to have red blood cells and you're going to produce these A markers and these B markers on their surface. I would say this is something to memorize. You should know which blood type is dominant to another blood type, which ones are codominant to one another. Obviously, if you have 2 alleles for A, you're going to have A blood. It's going to look just like the red blood cells of this individual, who is heterozygous for A blood, has a code for A and a code for O. This is going to be B blood. This individual is also going to have B blood, and it's going to look exactly the same as this individual that is homozygous for B blood. And the only way to have O blood is if you're homozygous for O. Again, blood type is an exception to Mendelian genetics because there's more than 2 alleles for a trait. All the traits that Mendel worked with, there was only 2 alleles for every single trait that he was looking at for pea plants. In addition, the A and the B alleles do not show the law of dominance because they're codominant to one another. The next trait that doesn't follow the ideas of Mendelian genetics is polygenic inheritance. For these traits, there's more than one gene that is leading to the phenotype of that trait. And again, Mendel was working with these pea plants, and for every trait, there was just one gene with 2 different alleles that was providing the phenotype for that trait. Well, here's a situation where there's 3 different genes coding for a trait. So with wheat seed color, there's 3 different genes that all lead to that final phenotype, whatever that wheat seed is going to have for a color. It says each dominant allele has a quantitative effect on the phenotype. So the more dominant alleles you have, the darker the seed coat's going to be. So this is going to be a very light seed coat. It doesn't have any of those dominant alleles. Over here, we can see that we have 6 dominant alleles. So this is going to be the darkest variety. Traits in humans that show polygenic inheritance would be skin color, eye color. And there's actually a really large number because most of our traits are just not determined by a single gene with 2 different alleles. Moving into sex-linked inheritances. So remember we said that most people have 2 codes for every trait. Well, that's not going to be the case for traits that are found on the sex chromosomes. Because if there's a gene on the X and guys are XY, then they're only going to get 1 allele for these traits that are found on the X because the alleles on the X do not match the alleles on the Y. They're not homologous. And I just gave you some examples of some genes we find on the X, and they're only on the X, they're not on the Y. And we look at these Y genes, they're not found on the X. So guys are only going to get 1 allele, 1 code for traits that are sex-linked. Females will get 2 though. But again, these males are not going to be following those ideas of Mendelian genetics. Because Mendel said you get 2 codes for every single trait. So that's why it's that exception. Males do not have 2 alleles for each trait, for these sex-linked traits. If genes are found on the X chromosome, sometimes we say they're X-linked. If genes are found on the Y, then we'll say they're Y-linked. If a gene is found on the X chromosome, then we're going to write the X, and we're going to put a letter on it to represent which allele is found on that X chromosome. If the gene is found on the Y, then we'll use a Y and we'll put a letter associated with the Y as well. A classic example of a trait that is sex-linked is color blindness. So the gene for color blindness is found on the X chromosome, and being able to see color normally is dominant. Color blindness is recessive. Since we know that this trait is on the X chromosome and that seeing color is dominant, if we have a heterozygous female with normal eyesight, you would have to know that her genotype would be X with a big B, X with a little b. She's heterozygous for the trait. Homozygous female with normal eyesight would be X big B, X big B. Homozygous colorblind female, you would have to know she has to have 2 codes to be colorblind. Now let's look at the genotypes for the males. Males with normal eyesight, he's going to have a code for normal on his X, and then he has the Y. He doesn't have 2 X's. A colorblind male has a code for colorblindness on his X, and he also has the Y. One thing to note about genes that are found on the X chromosome is that more males than females will develop that condition. And it seems counterintuitive because you think, oh, if it's on the X, well, girls are getting 2 X's. They definitely would more likely develop these traits that are associated with the X chromosome. Well, a lot of the traits are recessive, and because they're recessive, then they're not going to show up so often in the females, but they are in the males. And here's the reason why. Think about it. If a female gets 1 recessive allele from a parent, will she have the trait? No, because she has this one that can mask this one. So she's not going to show color blindness. If a male receives 1 recessive allele, he's got it. He does not have another X that could have that dominant normal allele that could cover up that recessive one. And again, there's no corresponding allele on the Y for this trait because the X and the Y are not homologous chromosomes. Let's go through a few sex-linked problems, and then I'm going to show you some that are really going to make you think. And these are typical AP problems. We have in fruit flies, eye color is sex-linked. Red eyes are the wild type and are dominant to white eyes. Now I'm trying to teach you some other terminology that you're going to see here in these genetic problems. If you see a trait and they say it's the wild type, I want you to understand it is the most common. So red is most common in fruit flies. That's the color of their eyes. That's what we normally see. This other phenotype that shows up on occasion, that's not very common, is usually the mutant. It's due to a mutation. And for this phenotype, lots of times we see a negative sign associated with it. So I'm going to put X negative, and that's going to be the symbol for white eyes in this situation. The wild type usually gets a plus associated with it. Now you're going to see lots of problems where they're talking about the wild type, and you're going to see genotypes that have pluses in them and minuses. So as we see more of these problems, you're going to get more comfortable with what that plus and what that minus means. So let's cross a heterozygous female with a wild type male. If she's heterozygous, she's going to have red eyes, and we're going to put this for her symbol. She's heterozygous. She has two different codes. A wild type male, well, wild type was red. So for him to have red eyes, we're going to put a plus there on his X, symbolizing red, and then he has the Y. So we bring him down and take him across. Now, since this is sex-linked, we're going to be reporting phenotypes with both what they look like in reference to their eyes, but also with the sex. So here I have two females, and 2 out of 4, they're females, and they're both wild type. They both have red eyes. I have one of these males, 1/4, male, and he's got red eyes. He's got that wild type. And here's a mutant male. He has those white eyes. The next question says, cross a purebred wild type female with a mutant male. So we're going to have purebred wild type female. Here she is, X plus X plus. A mutant male would be X minus Y. So she can pass on either an X with a plus and X with a plus. He can only pass on an X that has that code for white or his Y. So bring him down, take him across. For their phenotypes, we can report 2 out of 4, female. We can put wild type, or we can put red eyes. 2 out of 4, males, and they're also wild type as well. But again, when they're sex-linked, you have to denote the sex and then also what variation of the trait they possess. Now, this is where it's going to get a little bit difficult. Basically, what you're going to have to do in AP problems is, again, you're going to be shown the results of a cross. You're going to be shown the phenotypes of the offspring. And you have to usually work backwards. You have to figure out the genotypes of the parents. Before you can figure out the genotypes of the parents, you have to figure out what type of inheritance is going on. Is this trait sex-linked? Is it showing incomplete dominance, codominance? Is it showing complete dominance? So before you tackle these problems, there's really 2 decisions that you have to make. You have to take a look at the results here, and you need to make these 2 decisions. Now you're going to see that these decisions that we're making here are very similar to the decisions that we made whenever we were trying to solve pedigrees and figure out the genotypes of people in a pedigree. Remember the pedigree with the circles and the squares? The family trees, basically. Your first decision was, is the trait autosomal or is it sex-linked? And your second decision is this trait that's being mapped out, is it a dominant trait or is it a recessive trait? So these are the 2 decisions that we're going to have to make whenever we take a look at our results here. Before we go through and answer this problem, if you want, you can hit pause and you can see if you can answer A. Can you figure out the genotypes of the parents in cross 1? So to tackle this problem, first I note, ultimately, what are they going to have me answer here? What are the genotypes of the parents? In cross 1, we have a gray male, so I'm going to indicate I have to figure him out. And then there was a black female. And we cross those two, and this is what we get. And then we take a black male from this generation, and we're crossing it with a white body female, and this is what we get. Okay? So let's look at our first decision that we're going to make here. Is this trait showing complete dominance, codominance, or incomplete dominance? If the trait is showing complete dominance, then we have to figure out which allele is the dominant one. Well, I see gray, black, and white phenotypes in this problem. So that tells me that I'm dealing with a trait that shows incomplete dominance. So if I'm going to choose letters to write, I'm probably going to write like B, B prime would be my gray. I'm going to have B, B, B black, and white would be B prime, B prime. Now the next decision I'm going to make is this trait autosomal or is it sex-linked? So basically if it's autosomal, I'm just going to use regular letters like I already have written up there. If it's sex-linked, I've got to take those letters and I have to put them on an X or a Y. And I'm looking at this, and one way to tell whether it's autosomal or sex-linked is if you see one sex getting more of a certain phenotype. If you see that, then it's definitely sex-linked. If you see that these different phenotypes, it's just as common in males and females, then it's an autosomal trait, and I don't have to break out the X and Y chromosomes when I write genotypes. So looking at this, I have gray females and I have plenty gray males. I have black females, I have black males. I have gray females, I have gray males. And the numbers are pretty even. So that tells me right here that this is an autosomal trait. And basically it tells me I'm going to keep using these letters for these genotypes. I'm not going to do anything like this. If it was sex-linked, then I'd have to put those B's on the X or the Y chromosome. Okay, so jumping down to A, now that I have those two decisions made. What are the genotypes of the parents in cross 1? So first of all, since this is autosomal, I actually am going to try to ignore the male and the female part. And if you just look at this problem, it looks like I have the same number of gray organisms as I do black ones. Those numbers are pretty even. I have basically a 1 gray to 1 black ratio. And if I've done enough of these problems, then I probably have pictured a Punnett square in my head that would look like this. Gray is the middle, so gray is going to be the heterozygous individual. And then black, we said we would do this for their genotype. And if I do this to check if I'm right here, this is when I would get a 1-to-1 ratio. I'd get 1 gray, and I'd get 1 black. So I just figured out the genotypes of the parents. The gray male was B, B prime, and the black female was B, B. Just because we put the words female and male in here does not mean you should start busting out the X and the Y chromosomes and their genotypes. Only if we determine a trait is sex-linked do you start bringing out the X and the Y chromosomes in those genotypes. B says, explain how you could tell whether the gene for body color was autosomal or sex-linked. So if more of a sex develops a phenotype, the trait is sex-linked. If we see an even distribution of phenotypes between males and females, then the trait is autosomal. That's how we can tell. Now, it's unnecessary, but I'm just going to go on and show that I'm probably guessed right as to the genotypes of the gray male and the black female. Because if I go down to the second cross, it says a black male fruit fly from this was crossed with a white-bodied female. So in the second cross, this is what we're doing here. We have, says a black male, so that's going to be BB. It's crossed with a white-bodied female, so she would be B prime, B prime. then I would get all grays. And that's what we got. So all those offspring, 100% of them turned out to be gray. So here I have a old AP question set, and it's very similar to the question that we just finished answering. So take a second and read through the information. There's some multiple choice questions associated with it. And eventually I'm going to go through and I'm going to determine the genotypes of all the individuals of these crosses that you see here in the box. So if you want to try this on your own, go for it. Looking at the question prompt, it says, in a mating of a male, this is a fruit fly, collected in nature with a normal female from a laboratory strain, we ended up with this many offspring. It says all the offspring were normal, except one unusual male, and it had very narrow eyes. Now, the fact that you have 1 out of 237 should scream the answer to question number 1. The most likely explanation for the appearance of the narrow-eyed male in that P1 generation is because of a mutation. It says a series of matings is shown in the table below. Determine the genotypes of the individuals in the crosses. So they took this very unusual male and they crossed him with some normal females. There he is. Remember I said you need to make 2 decisions. First decision to help us analyze this data is this trait showing complete, incomplete, or codominance? And I've got normal eyes, so let's indicate I've got normal eyes. And I have slightly narrow eyes, so let's put slight. Then I have very narrow, so I'm going to put very narrow eyes. And this should signify that we've got incomplete dominance going on because there's 3 different phenotypes, and one is the middle of the other 2. It's going to be the one that's heterozygous. So I'm going to go ahead and assign some letters. It's hetero, incomplete. I'm going to put N prime. You can use whatever letters you want. So normal would be capital N, and the very narrow eyes, N prime. Second decision, autosomal or sex-linked, because I need to know if I need to leave the N's like this or I have to put them on X's or possibly Y's. If you notice here, see how all the males have a certain phenotype and all the females have a different phenotype? Then this is definitely sex-linked. So this tells me when I go to write the genotypes next to a male or a female, then I'm going to have to do things like this. I'm going to have to put like an X with an N on it and a Y, or something like this for the females. So let's go through and see if we can figure out the genotypes of these different individuals. So we have an unusual male. Remember, he had very narrow eyes, and I already kind of established the letters that I want to use. Since this is sex-linked, then I'm going to give him an X. I'm going to put an N prime, these are N primes, on his X, and there's a Y. The normal female, X capital, X capital. Now, to check this out, if I'm guessing right about the parents here, I'm going to do a cross to make sure this is what I'm going to get for their offspring, if this is in fact the genotype of the male and the female. So I have an X N prime Y. I have an X N X N. When I do my cross here, X N prime, X N Y, X N prime, and X N Y. So I should see all of the females, right here and right here. I should see all the females with a middle phenotype, with that slightly narrowing of the eyes. All the males should have normal eyes. Remember that capital N there with no prime indicates normal, and that checks out, so I'm on the right track. From that point on, I would probably stop and wouldn't work any more Punnett squares, and I would start answering the multiple choice questions now that I have a really good idea of what's going on here. So let's go down to 2. It says, assuming that a single locus causes the eye characteristic, so it's not a polygenic trait is what they're telling you there. The gene appears to function as, we already figured this one out, it's showing incomplete dominance. From the breeding data, which we have because we've worked through a Punnett square there, It is clear that the gene is physically located on what chromosome? Most of the time, it's going to be on the X if it's sex-linked. I'll eventually show you a problem where we see a trait that's on the Y, and that's pretty rare though. But you would also be able to tell it's not going to be these because there's no information about chromosomes in the question prompt. So you could probably get rid of 2, 3, and 4 pretty easily there. Question 4 says, why did the F1 females not show the genetic trait? So we might have to work out another Punnett square here. So let's take a look at those F1 males that are normal. Well, we know that they're this. They're the F1 generation because we took the normal males here and we crossed them with one of these females here. So this normal male is X and Y. Let's go down and look at the answers here. You've never seen this word before, never select it. There's no definition of this word in a question prompt or anything like that, so don't select it. You have to trust that I've taught you all the different ideas that I need you to know for the AP exam, and that's not one of them. The effect of the gene is blocked by the Y. Well, that would mean that the Y and the X are homologous and that there's an allele on one that can cover up the allele on the other. We know that's not the case. The allele is blocked by the dominant allele. This is incomplete dominance, not complete dominance. The trait is expressed only in alternate generations. I'm going to cross through that because we actually see the trait in all those generations. So we see normal at the very first generation, the P1 generation. We see normal in the F1 generation. We see normal in the F2 generation. So we're not seeing it over alternate generations. And simply this, they do not show that narrowing because this is what they are. They have that normal allele there. They do not have allele for those very narrow eyes. Question 5 says, a promising approach to determine if a female with very narrow eyes could be obtained would be to, well, let's try to figure out how we would get a female with very narrow eyes. She's going to get an X from dad. You got to have those X's with those narrow eyes. So we would have to cross a male that has very narrow eyes. And remember, we're trying to get a female that has very narrow eyes, and we don't know if we can. So that means that probably all we can have to cross with this male is a female that has those slight narrowed eyes. There's a slight narrowed female. So then we can bring them down, take them across, but you can see I'm going to get her right there. This would be a female that fits that genotype, has very narrow eyes. So let's look at our options. Cross the normal F2 males with the normal females. Nope. Cross the F2 males that are not normal with the females that are not normal. And that's an option. This is your not normal male with females that are also not normal. So I'm going to kind of star this one, see if there's anything better. Cross the F2 very narrow-eyed males with the normal. Nope, we need our female to not be normal. Cross the F2 slightly narrowed-eyed females with the normal males. Nope, we need our males to be abnormal. Back cross the F2 normal females. Again, we need them to both show those abnormal phenotypes. So B is our correct answer.